Okay, well, good morning, everybody. It's my great honor to introduce the Slatopolsky lectureship for you today. Dr. Slatopolsky, here in the first row with the mustache, is Ed, who was truly a true triple threat, like our speaker today. He made remarkable contributions to the teaching of generations of academic nephrologists and scientists that carry on his legacy today in their own research laboratories. He made groundbreaking discoveries in the areas of PTH, vitamin D biology, chronic kidney disease, and metabolic bone disease. And his discoveries have, in fact, improved the lives of our patients who suffer from kidney disease. In this renal division photo from 1981, we can see Dr. Slatopolsky in the front row, next to Salo Klar and Mabel Perkerson. It's hard to believe, but Dr. Slatopolsky had already been on the faculty for more than 15 years at this time. This is Dr. Slatopolsky's famous rooster named Macho. This particular rooster was a mean one, hence his name, but it produced the most potent and specific antibody against PTH the world had seen and enabled countless laboratories to measure parathyroid hormone levels for the first time. In fact, we still have Macho in the renal division. Not kidding. It's not an exaggeration to say that we in nephrology viewed Eduardo as Superman, but also appreciated his wonderful sense of humor and panache. We are all humbled and grateful at Ed's remarkable generosity to the division and school as well. He established the Eduardo and Judith Slatopolsky Professor of Medicine and Nephrology, and in 2022 we were able to celebrate Dr. Jeff Miner here in the audience as the inaugural incumbent. Dr. Slatopolsky passed away a year and a half ago here in Barnes, having attended renal grand rounds in our division just the week before. And so he leaves a tremendous legacy, and this lecture is named in his honor and his memory, and it is an equal delight and pleasure for me to introduce our speaker today, Samir Parikh, who received his undergraduate degree in chemistry from Harvard. He received his medical degree from Vanderbilt, and then completed internal medicine training at Beth Israel Deaconess, followed by nephrology training, and started on the faculty at BI. And he and I were compatriots back in Boston back in the day. He rose all the way to the rank of a professor, and in 2021 he was recruited to UT Southwestern, where he currently serves as chief of the Division of Nephrology. And Samir has made, as you will hear, truly important discoveries around acute kidney injury, chronic kidney disease, and the role of metabolism that have important translational and clinical impacts on patients suffering from kidney injury. Samir has in addition to his scientific contributions, has a truly laudable history of mentorship at all levels, from undergraduate to graduate to fellows, and already in his short career has a legacy of folks who came from his lab and who are starting their own groups. He is a member of the American Society for Clinical Investigation, the American Association of Physicians. He is on the Executive Council of the American Society of Nephrology and will serve as our 62nd president in 2 years' time. And he is just a terrific person, and we're so pleased to have you and welcome you here to Medical Grand Rounds today, Samir. Now let me just pull up this talk. Here we go.
Well, thank you. How's my audio? Is that okay? Oh, wow. It's fun to have a group like this here. I really appreciate you making the time to come down. Thank you, Ben. Thank you to the Nephrology Division for the opportunity to be here with you today. I know it's so much easier to click on a Zoom link than it is to show up in person, so I appreciate the community that we have here. Okay, so first off, a hello from Dallas. This is the picture on the left is our annual Halloween party, and it's just coincidence that I happen to be dressed as one of your cross-state celebrities here in Travis Kelce. I'm trying to see who can you pick out here who's Wash U. So this is Hani Suleiman from the Renal Division, Eva Langner, and Yoshi Muto and his wife, all recent recruits from your amazing division to our faculty. I also wanted to share that for me, this is very much a homecoming. As Ben mentioned, we grew up together in Boston, and I see Ben as a big brother. He taught me how to do kidney injury models, and how much fun it was to celebrate AAP induction over here in the middle. Ben has served on a national panel that we're both involved with to promote kidney research and more funding for kidney research. And I've had the pleasure to work with Jeff on the council at the American Society of Nephrology, which Ben is joining in short order. So thank you again. This is very much a welcome home for me. These are my disclosures. None of these are really relevant to the contents I'll present, but just to be thorough. And I want to start at the end by acknowledging the people whose contributions I'll be sharing with you, and I'll try to have photographs all throughout so that you can appreciate the people who've come through. The funding, of course, and I only had the pleasure of meeting Dr. Slatopolsky once, and it was in a social setting. As all of you know, he's not a tall man, but he really owned the room. Wherever he was, his presence was larger than life, and so much joie de vivre. We tried to do a little bit of that last night at the brasserie. We enjoyed some wine and good food, but it would have been much lovelier to have Dr. Slatopolsky with us. So I'm deeply honored. I remember learning the trade-off hypothesis when I was a fellow, and that's how I teach secondary hyperparathyroidism to my fellows these days. So an enormous legacy that you have here. Okay, so this is medical grand round, so we have to have learning objectives, and these are vague. They're not sort of typical learning objectives, and so I apologize for not using the right words. So here's what I'd like for us to achieve in the next 30 to 40 minutes. Number one, appreciate acute kidney injury as an event that happens between host and stressor. And I'll get into this a little bit more. I was speaking with one of the chief residents who's going into palliative care. AKI is really an event that happens in older individuals. It is probably one of the strongest age-associated conditions out there. And that question, why is that, has really prompted a lot of our research. Second, I hope you walk out of here with some appreciation of state-of-the-art management of acute kidney injury. That's the practical stuff. And then finally, I'd like to share and convey some experiments of nature, as well as some of the work that we and other groups have done that hopefully help you think about the renal tubule as a machine of sorts. If you don't already think about it that way, I'd love for you to think about it as a machine of sorts. And this very typical measure that we do for every acute kidney injury patient, the FENA, is really a measurement of that metabolic capacity. Okay, so again, since this is a medical grand round, start with a case. This was actually, early on in my career, we were asked to see a young man, a very young man, who was admitted to Beth Israel Deaconess in Boston. He was a senior at Brookline High School, and he was the captain of the soccer team. He had recently had EBV and had been on bed rest, but because he was the captain and it was the summertime, he felt like he had to join for 2-a-day practices. And you can see what that, the intensity of the practice here written up. And he was admitted with diffuse muscle weakness and soreness, no significant past medical history other than the EBV, and the exam was unremarkable other than tenderness kind of in all the major muscle groups. So the labs were notable for a CK of 225,000. Is the diagnosis a mystery? Does anyone want to shout it out? Rhabdo, absolutely, rhabdo. So rhabdo was the diagnosis. But what made this a bit of a curveball was that the question to us was, this person does not have acute kidney injury. What do we do with that? Because if you have acute kidney injury, the management is very simple, right? You give isotonic fluids, you promote a urine output of close to 200 cc's an hour. If you want your note to sound erudite, you might pontificate on whether that should be a little bit more base or a little bit more acid. And if you want it to sound even more erudite, you might mention something about clearance of myoglobin by filters for dialysis. But really, it's quite simple, right? Volume expand, make him pee out the myoglobin. But this was a curveball because he didn't have acute kidney injury, and they were asking, well, can we just let him go, or do we keep him in-house and keep sort of flooding him with fluids? And that case gave me an appreciation for negative space. And this is what I mean by negative space. I have a couple of pieces of art up here, one of them from your wonderful museum, the St. Louis Museum of Art, that I got to see at least the outside of yesterday evening with Jeff. What's not there is often as important as what is there. And I remember during fellowship, I had a moment where of deep gratitude that I wasn't being consulted on every single post-cardiac surgery patient. You know, thank goodness it's not every ICU patient. Thank goodness it's not. Because we do remarkable things to our patients, right? And so what's not there in this young man's case prompted a lot of thinking on my part, at least. So acute kidney injury, I already mentioned, at the confluence of stressor, different kinds of stressors, and the at-risk population. One could imagine in a thought experiment that the readout has a distribution, and it probably is not a normal distribution like this. But on one end, the high serum creatinines, we know who those people are, right? They're diabetics, they have chronic kidney disease, they may have heart failure, they are definitely older. But what about the other end of the distribution? Who survives stressor and gets through it, like this young man, this huge CK? And what is the nature of that resilience? And that's kind of been the question we have focused on for well over a decade. So why study acute kidney injury? The reasons on the left and right are, I would say, the medical reasons, right? It is common, super common. I don't think you can have practiced a day of inpatient medicine without having seen an acute kidney injury patient. 20% of adult hospitalizations, very costly length of stay. A significant chunk of it fails to resolve fully, and so now you're left with chronic kidney disease in its wake, and chronic kidney disease is super common in this country and around the world. We're talking about 1 in 10 people in the US and nearly 1 billion people worldwide who have chronic kidney disease. So very impactful at the level of patients and populations. And then on the right-hand side, acute kidney injury is an orphan condition, right? The only thing we really have to offer, and we'll talk about this, is dialysis. But one could imagine that if a treatment was available, it could scale from acute kidney injury into the chronic kidney disease arena, and so it could have big impacts. So our question, what drives resistance to and recovery from acute kidney injury? And then the working hypothesis that I'm going to tack towards as we go through this is that mitochondrial function may provide an interesting window to look at this. I feel a little bit funny talking about acute kidney injury here because your chief of nephrology has published, maybe a paper or two in this arena. Ben has been not just prolific, but highly impactful in his career. Leslie Ewan, as well in the audience, has done outstanding work in this arena. So you'll correct me when I say wrong things in real time. This is a very common disease, as I already mentioned. On the left-hand side, you can see some of the epidemiology from the national inpatient sample. On the right-hand side are death and taxes. How do you measure what kind of change in serum creatinine matters? And this cohort study that Glenn Chertow conducted towards the end of his time in Boston before he moved out to the West was to figure out that even modest changes in serum creatinine matter. And the two outputs that he measured were death and taxes. So the top graph, the bar graph on the right-hand side, let me make sure I've got my mouse here, is measuring stepwise serum creatinine changes as they grow in magnitude and what are the odds of death. And then this lower graph is measuring the amount of money back then, 2.5 decades ago, that was being spent as a function of serum creatinine changes. And so, sometimes I think we think of AKI as, well, that's a numerical abnormality and what's the big deal? But the epidemiological data are very strong and very clear on even modest changes are important. And I guess the question I'll put in your head at this juncture, if you haven't already considered it, is acute kidney injury a kind of failed stress response, right? That it's something that unmasks a problem that the host already had? or is it an event that sort of changes the future trajectory of kidney health and total body health? And I'll just put that question out there. I think the answer is both, but we'll try to get there. So today, what do? If you're a house officer or a fellow here, we stage AKI by looking under a microscope and by doing some functional tests, right? And I put two of the functional evaluations here. The FENA we'll come back to. I'd love for you to think about the FENA not as a cutoff of 1% or not, because all of us in this room should be at a FENA of about 1% or lower, unless you had a bunch of nachos last night or what have you. But none of you have acute kidney injury. Rather, I'd like you to think of a low FENA as your metabolically active machine in the tubule working very nicely. In addition to structure and function, we're trying to get to molecules. We're not there yet. People have been talking about these for 2+ decades, so I won't belabor this. In terms of management, this is a very busy slide. The one tool that we've had, and we've had since World War II, is dialysis. And dialysis obviously saves lives, and we do a lot of dialysis. During COVID, we were running out of dialysis machines. We were running out of fluids to hang on the CRRT machines. But when we look at the trial basis, the evidence that's out there for can we affect the course of patients by manipulating this tool we have of dialysis, the answer is frustratingly no. And so these are studies, some smaller, some larger. I'll point this one out, START-AKI, nearly 3,000 patients. And they all have tested the hypothesis of early versus late initiation. Can we move the needle on outcomes? And the answer is no. And the answer has been no again and again and again. And that's very frustrating for us because we'd obviously like the answer to be yes. We'd like to be able to impact these outcomes. Pushing the limits of this platform of dialysis, of a cartridge that you run the blood past. These results were just announced by a company collaborating with Baxter. This is John Kellum, for those who know, in the renal and critical care fields. John took a sabbatical to work with this company. This is a very old idea, and the old idea is to impregnate an antibiotic on a cartridge and then have the blood run past that cartridge, in this case to treat endotoxic shock and clear the pathogen by heme absorption, and thereby see if they can get better outcomes in septic shock and sepsis-associated acute kidney injury. The results are not out yet. This just was announced in August, and so it may be coming out at our upcoming Kidney Week meeting. But this is really trying to push that platform of dialysis or extracorporeal therapies to its maximum. And so before we leave the world of clinical trials and clinical management in AKI, if you think about where to conduct trials, where on the patient journey we should be conducting trials, there's a rationale to work very early on this spectrum, like primary prevention of acute kidney injury, but there's also a rationale to work very late, and I'll just briefly put those up. The primary prevention stuff is nice because it's a controlled insult. You know when it's going to happen. So for example, a cisplatin AKI trial. Cisplatin doesn't wind up in the body as a surprise. You know when it's going to happen, so it's easy to design a prevention study. Cardiac surgery is a very common grounds for phase 1 and phase 2 studies. And so it's the controlled insult, it's easy to conduct. However, the event rate is really low. You know, it goes back to this point of resilience. Most of the time, we don't get into trouble. Even though AKI is super common, we don't get into trouble all the time. So the event rate is low for these kinds of primary prevention trials. And the real world application, not as evident, right? We really descend on those who do get into trouble. And so there's also a rationale to be on the right-hand side of this patient journey. The significance of the endpoints, they matter, right? I mean, hard endpoints like body counts matter, the effect size, and then the real-world application. I'll give you an example of a study that was on the right-hand side of this distribution. This is the Revival trial, which is a phase 3 study conducted on an inhibitor of alkaline phosphatase. among individuals with sepsis and acute kidney injury. Relatively small study, about 360 patients, 1-to-1 placebo to active. Their primary endpoint was a hard endpoint, 28-day mortality. And their secondary endpoint was also a hard endpoint. MAKE stands for Major Adverse Kidney Events. So that would be things like persistent dialysis or persistent doubling of serum creatinine, i.e. Chronic kidney disease, at the 90-day mark, so MAKE 90. And what you can see here, whenever lines cross, that's a bad thing. That's what I learned in my statistics class in med school. So the intervention didn't work, right? So this is really frustrating for us. Primary prevention trials, trials with existing tools like dialysis, trials with new agents along the entire spectrum, and we don't have innovation that we can bring to patients' bedside, not yet. So with that, I'm going to strike a pivot into the world of research and have you kind of think along with me, and I welcome your input because this is very much in media res, and you know, I always learn from groups. And so we'll talk a little bit about this concept of a metabolically active machine. And I'll tell you a bit about how we've tried to take that machine apart. And if I do the job well, we can also talk about this question of, is acute kidney injury a failed stress test? Or does it set the organ on an adverse path? So let's start with some comparative physiology. Kidney researchers love this stuff. I think it's an excuse to like go fishing or hang out by a coast. I'm in Dallas, you guys are in St. Louis. We don't have big coasts to hang out in. So when you look at marine organisms, they balance the tonicity of their blood by getting rid of the hypertonic fluid that's coming in, right? Seawater is 3x the tonicity of blood, and so they've got to pump sodium out in order to maintain the correct saltiness of their blood. And to do that pumping, they need mitochondria to generate the energy to do the chemical work, right? If you switch that story to land-based creatures, we've got the opposite challenge. We are consuming hypotonic all the time, right? Even when you have Gatorade, that's still hypotonic. And at least evolutionarily, we didn't have a ton of sodium in our diet, and so we had to retain sodium. And not to drag people too much into physiology, but how you retain water in the body is by bringing the sodium back into the body, right? When you think about the filtered load of fluid at the glomerulus, that's 100x higher than your urine output. It's 100x higher. So how do you get 99 waters to march back into the body? Well, the way you do it is to pump sodium back into the body, and the water follows along, right? That's sort of the big lesson from med school renal physiology. So mitochondria is still important, sodium transport is still important, but the vector points in the opposite direction. Instead of the vector pointing out, it points back into the body. Okay. And there are adaptations to make this happen in terrestrial organisms. One adaptation depicted on the left. The cells that are responsible for that sodium movement are essentially bags of mitochondria. Right? They're essentially bags of mitochondria. The mitochondria are all of these electron-dense, sausage-looking things that have been cut in cross-section. That's one adaptation. The other adaptation is to massively increase the surface area through this beautiful apical architecture of microvilli. And I love this scanning EM photo because you're looking right down the barrel of the proximal tubule, and it looks like a shag carpet. And so you can imagine all that surface that's available for all that sodium transport. And then you have these mitochondria generating the ATP that then works on the basolateral side to kind of create a sodium vacuum, to have that sodium moving right back into the body, right? So that's this sort of how I try to teach it to our fellows. So it's a lot of chemical work happening in the renal tubule all the time. We reabsorb about 1 pound of sodium in the renal tubule every day, and we do that 1 pound atom by atom. And so there's this kind of quiet chemical work happening all the time in the renal tubule. There are more lines of evidence to suggest that mitochondria may be important. Experiments of nature. If you look at individuals, rare individuals who have mutations in genes that govern mitochondrial structure and function, tubulopathy, which I'll argue to you is a kind of slow motion form of acute kidney injury, a number of the same hallmarks. Tubulopathy is very common. More than half of individuals who have mitochondrial mutations have tubulopathies. And then if you think about the stuff we do to our patients, there are a number of interventions. The first class of diuretics, for example, heavy metals, these wind up replacing the iron and iron sulfur clusters that are in mitochondria. Nucleoside RTIs, reverse transcriptase inhibitors, poison mitochondrial DNA, aminoglycosides, poison mitochondrial function. And all of these things are associated with tubulopathy. And so one of the challenges in sort of studying acute kidney injury has been, well, there's no genetics of it. There's no, oh, here are these rare individuals who have, profound AKI, and can we go rare to common? But I think part of the reason why there hasn't been a genetics of it is, well, this is very evolutionarily undesirable, right, to be super susceptible to acute kidney injury. But I think we've also not been asking the right question. Okay, so here's the hypothesis. A normal healthy tubule on the left doing all of this vectorial sodium transport, various hits come along, and now the mitochondria don't work, and sodium transport kind of fails, and the whole function kind of collapses. In a nutshell, that's the idea. All right. So with that, let me sort of take you through some of the work we've done to test this idea. One of the joys, I suppose, of working in experimental systems is you can manipulate the variable you're interested in, right? We manipulated mitochondria in mouse systems by manipulating this gene PGC1 alpha. The gene really doesn't matter, but the outputs of it do. And so PGC1 alpha is important for increasing the abundance of mitochondria in cells, and it increases the turnover. So it gets rid of kind of bad mitochondria and replaces them with healthy mitochondria. And so over a series of papers, this is in the previous decade, we tested different models of acute kidney injury with gain of function for this PGC1 alpha, so we kind of supercharged the mitochondria, and with loss of function, right? So we kind of completed a matrix of experiments where we looked at different kinds of insults, infection, ischemia, toxins. We looked at knockouts, and we found across the board, no matter the insult, the knockouts were more susceptible to stress. And we looked at the opposite, gain of function. So we just put more of this PGC program back into renal tubules, and across the board, they were more resistant to the stress. So that sort of fit with this idea that if you could fortify mitochondria, you could potentially fortify the response to the stressor. So we dug in a little bit deeper to think about mitochondria, and I apologize because it's only a little after 8, and I'm showing you biochemistry here. But how do renal mitochondria generate ATP and energy? The chief fuel source is fatty acids. Renal tubular epithelium can use other fuel sources, but fatty acids are the primary fuel. And the output is ATP. Down here depicted the various complexes we all had to memorize in med school or grad school. And in the middle is this molecule NAD. NAD is responsible for carrying those high energy electrons from metabolism down into the oxidative phosphorylation. We did a bunch of chemical surveys on these kidneys, and we found that in acute kidney injury, the precursor of fatty acids started building up. So fuel was building up, and at the same time, ATP was a lot lower. So fuel's building up, the output of the fuel is not showing up, and in the middle, this NAD was lower. We found that those knockout animals that were very susceptible had the same pattern in their kidneys at baseline, the ones that are really susceptible to all different kinds of stress, as opposed to the ones that were very resilient against the stressors. They had the opposite pattern. Hardly any fuel sitting around, a bunch of ATP present, and more NAD than normal. And so that was interesting to us, and we wanted to dig in further into how these little batteries were working in these cells. And again, I won't take you through the full details of it, but what we found was a series of enzymes that are responsible for generating NAD inside cells were all moving as a group. They were all falling in acute kidney injury setting, They were all down in these highly susceptible kidneys at baseline, and they were all up in these highly resilient kidneys. And I'll show you in just a few slides how that wound up sort of telling us something about people who face these stressors. So again, some more biochemistry, and I apologize. The pathway converts these enzymes, IDO down to NMNAT, sequentially convert tryptophan, essential amino acid, down into NAD, a series of 8 steps in the middle. And the kidney is one of the only places in the body that really uses this pathway with significant flux. And so just to pull back out for a second, the analogy that I sort of carry in my mind is if you've got an engine and the engine has to do some combustion and make some outputs to get the thing moving or working, that's really mitochondria, and the output is ATP, and that gets the proximal tubule or the renal tubule working. And this NAD in the middle, it's not quite a catalyst, but you can think of it like a catalyst. You need enough NAD around to kind of drive this whole process. One of the joys for me has been after we put our, our ideas out there, many different groups around the country and really around the world have verified and extended this concept in different directions. And I'll let you just look at those citations for a second. And it continues to sort of build on itself. So here's part of the human application. It's very evolutionary conserved, this pathway. And when you start at the beginning, tryptophan can become like a bazillion different things. Only about 10% of tryptophan winds up becoming NAD. But by the time you get all the way down to the lower part of this pathway, to this quinolinic acid, which is an intermediate, the only metabolic fate of quinolinic acid that we're aware of is that it becomes NAD. That's it. There are no other arrows that come off quinolinic acid. So we took that idea and reasoned that, well, look, if there's a block in this pathway towards the end, could there be a buildup of quinolinic acid? And what we found was absolutely there is in people. And so this study was conducted by Ken Ralto, who's now on the faculty at UMass, an associate professor. Ken looked at, in collaboration with colleagues across the street when we were in Boston, looked at a cohort of individuals admitted to the intensive care unit. About a quarter of these people eventually developed acute kidney injury. And what Ken found was that when he looked at quinolinic acid levels in the urine, it dose proportionally predicted who would get AKI, right? So it was antecedent. The rise in quinolinic acid was antecedent to the event of AKI. So this gets back to the idea of who's ready to handle stress and who might not be ready to handle stress. And there were multiple sort of statistical models that he and the team worked through. But no matter sort of what adjustments they undertook, this dose proportional relationship persisted. And you can see what the odds ratios were like on the left-hand side. Very powerful. This concept that high levels of urinary quinolinic acid, indicative of a block in the body's ability, or a deficiency in the body's ability to convert tryptophan to NAD, this was independently verified by a European group who looked at 2 other settings. So ICU up here, this is coronary artery bypass grafting. We talked about cabbages a little bit earlier, a setting that triggers acute kidney injury. They found that the urine quinolinic acid levels in their patients were more predictive of a future diagnosis of acute kidney injury than one of the best markers out there called NGAL, molecular markers. They also looked at another setting where we see a lot of acute kidney injury, which is right after kidney transplant. New kidney goes in, it's been ischemic for a while, it's been sitting on the table, it's been in a Coleman cooler, what have you. It goes in, it doesn't work immediately. The transplant nephrologist called that delayed graft function. That is also a kind of acute kidney injury. So we think that we can peripherally measure how all of us in this room might be different when it comes to throughput on this metabolic pathway and our readiness to handle stress. But measuring is half the problem, right? The other half is intervening. And so here we took advantage of the idea that there are multiple ways to make NAD in the body. And this tryptophan pathway, which I'm now showing in yellow, is one way to do it. That's the way we saw that was being kind of manipulated by this mitochondrial gene called PGC1 alpha. But the much more sort of efficient pathway to make NAD is down here in the green. This is from nicotinamide, or vitamin B3. And so this guy, I want to do this at some point in my life. This guy published a paper in Science that was a one-figure paper, Conrad Elvegem. I think it had 3 rats in it. And I think for this paper and other work, he won the Lasker Prize a few years later. And what Dr. Elvegem showed in this paper was that a disease that's associated with low levels of vitamin B3, that's pellagra, could actually be treated with a bunch of tryptophan. And so what he was showing in this paper was the fungibility of NAD precursors. Does that make sense? Pellagra is a situation where you don't have a lot of vitamin B3, this thing in green. And in this paper, Elvegem's group was showing that you could give a whole bunch of tryptophan and start addressing the problem of pellagra. Conversely, if you start out with an issue with tryptophan, that's called Hartnup disease, where you've got a problem with neutral amino acid transport, the treatment for that is to give a whole bunch of nicotinamide. Okay? And so in both directions, this is fungible. And we've taken advantage of that because we think that in acute kidney injury, the deficit in the kidney, one of the important deficits is on this yellow pathway to making NAD. And we've said, well, look, if these guys thought that it's fungible in both directions, let's try to sneak in with nicotinamide or B3. Does that make sense to people? Maybe, sort of. And here's what that looks like. 5 trials out there. So we published the original sort of mouse data in 2016, and these trials have come out, you can see, 2018 and ongoing. And they're in different settings. We talked about cardiac surgery early on. We've talked about ICU-associated AKI. COVID-related acute kidney injury, not the rare kind that leads to a ton of proteinuria, but the super common kind where people were coming in very sick and couldn't breathe well and had shock and they were getting acute kidney injury. And two now faculty, but started when they were fellows at different points in their career, Ali Poyenmer, who's out on the West Coast and has an R01 to do this study down here of 360 patients, and Nate Rains, who's on the faculty at BI, now did this COVID AKI study. Here's what they have found. And they're not the only ones who've been working on this. There are a couple of other groups, one in South America who've looked at this question, and another group in South Asia who've looked at this question as well. We're waiting on the results of this 360 patient study. There's a prevention design, so I already shared with you what I think some of the problems are with that, namely that the event rate is small, so Ali is trying to use molecular markers to work around that. But it looks pretty promising. And obviously our goal, could we deliver? I mean, vitamin B3 is something you can go buy at your local pharmacy. And so does this open up a way towards effective, safe, cheap therapy? And if not, nicotinamide, something that's sort of metabolically inspired. Okay, and so before I leave this section, let me just walk you through one experiment of nature. You all learned about pellagra. And the way I learned it was the 4 Ds, dermatitis, diarrhea, dementia, and death. So if everything I've said to you is true, how come no kidney phenotype with pellagra has been described, right? And Gaspar Casal in Renaissance Europe described this cutaneous lesion, the Casal collar, or the mal de la rose on the neck as a cutaneous manifestation of pellagra. The US has a history with pellagra. In the American South, there were pellagra hospitals in the 19th and 20th century, early 20th century, as diet sort of shifted towards corn and the proper treatments weren't done in a corn-based diet to extract the vitamin B3. And, I love looking at really old literature. And it turns out that, this is a very complicated y-axis here, but the nephrologists and renal researchers will recognize that urine creatinine excretion on a daily basis is kind of a proxy for kidney function. And What this group found and reported in the late 1960s was that they took children with malnutrition in sub-Saharan Africa, treated them with a whole bunch of nicotinamide over the course of several weeks, and they shifted the curve. They didn't report out that they improved kidney function. They just reported out that urine creatinine went higher. And this was not a result of just gaining body mass because creatinine is produced in the mass, because if you divide through by the body mass, the relationship still persists. And so this is the only evidence I've found that in a malnourished state, nicotinamide can enhance kidney function. So maybe a 5D will, with more studies, turn out to be dialysis. Last experiment of nature, this is now genetic. A couple of years after we published our first work in Nature, this study came out in the New England Journal from Sally Dunwoody's group, who is a pediatric researcher in Australia, New Zealand. And she looked at patients who have rare mutations in two enzymes, one of two enzymes in this pathway converting tryptophan all the way down to NAD. She looked at this Kainu enzyme and this HAO enzyme, loss of function mutations in families. These mutations are associated with multi-system congenital abnormalities. But what she showed was that the renal abnormality, which is, where is it here? Small kidneys, or renal agenesis or renal dysplasia. When you model it in an experimental system, it can be reversed by providing the pregnant mom with NAD precursors. And so these data argue that in people, deficits in the ability to produce NAD leads to renal consequences, right? Genetically, in a very targeted way. And so just to summarize all of that, here's where we're at. We think that this pathway may yield some new kidney injury biomarkers that may speak to the resilience of the kidney. And we think that this may point the way to some potential treatments, either to prevent or to treat acute kidney injury. And it's nice that to have buttressing from true experiments of nature, whether those come from the genetics arena or nutrition. So in the last couple of minutes, let me just tell you a bit about memory of kidney damage. I already mentioned that acute kidney injury is very common as you get older. If you think about the overall rate of hospitalizations, that goes up by a factor of 10 when you move from childhood into the 8th decade of life. So that changes by a factor of 10. If you look at acute kidney injury, that changes by a factor of 5 just in the last couple of decades in life. So even among age-associated conditions, AKI is on an extreme. And it's so common as a function of aging that once you're 85 plus, you're looking at a risk of acute kidney injury that's 1 in 10 per year. That's a lot. My father's 85, and so I shudder to think that he has a 10% chance this year of getting acute kidney injury. There's a relationship between acute kidney injury and chronic kidney disease. We've already talked about partial or unrecovered acute kidney injury as being a way that AKI actually leads to CKD right there from the event. We can also think of AKI or small AKI events as really degrading kidney function. This is particularly true for like heart failure patients, right? They come in, they get diuresed, they leave with a serum creatinine that's a bit higher, and it never comes back down to its previous baseline. And they come in the next time and it just sort of keeps ratcheting up. Its own little trade-off hypothesis. And then of course we know that there's an age-related decline in kidney function. Peak kidney function happens around the age of 30, and then we're all kind of losing about an ml per minute per year with significant variation. So what is all of that? How do we sort of put all of that together? Here are a couple of papers from Science, one in the 90s and one in the 2000s. Giuseppe Attardi on the left looked at people and measured in their blood that among the hallmarks of aging was the acquisition of mutations in the mitochondrial DNA. So to remind this group, we have nuclear DNA, 2 copies in each nucleus, but we've got tens to hundreds of copies of mitochondrial DNA inside every cell. So we're polyploid for mitochondrial DNA. And Attardi's group described the acquisition of these mutations as a function of aging, so somatically acquired mutations. Then this group came along, Proya's group, and said, well, look, why don't we do a sufficiency experiment? So they took mice and they introduced a mutation that would lead to a bunch more mutations. So they mutated this gene called polymerase gamma, which is responsible for replicating mitochondrial DNA. And the mutation they put in this pol gamma was one that screwed with its proofreading ability. And so these mice had a very rapid rate of mitochondrial DNA mutation acquisition. And you can just see from the pictures, they looked old when they were young, and they wound up dying at a much earlier time point than their controls. And so we, led by Vivian Huang, who's now on the faculty in Boston at Harvard, have thought about, as we peel the layers back on this machine, inside the machine, or the battery inside the machine, could there be information degradation happening in the way that these batteries work? And what Vivian found was that injury, this sort of transient injury, seems to lead to permanent mutations in mitochondrial DNA, and that these permanent mutations make the kidney very frail to future insults. These mutations arise not just from injury, but they also arise as a feature of aging, and they are probably also influenced by the variants that all of us have inherited that control the rate of appearance and disappearance of these mutations. So genetic component, aging component, and an injury component. Here's what her work looked like. On this x-axis, if you will, I've unwound the mitochondrial genome from position 1 to position 16,000, what have you. And wherever you see these little white lines, those are mutations. When you read and reread and reread sequences, you can pick up low-frequency mutations. Each row is a kidney. Sham-operated, IRI is a kidney injury model, looking at it at an acute time point, so 24 hours after the insult, and then coming back and looking at it a month later. The big surprise, well, the two big surprises was, one, there's this rapid burst of mutations that happens and visible within 24 hours. The second surprise was that they persist. Even after functional resolution of AKI in these animals, the mutations persist. They just sort of sit there. I didn't really believe this work until we collaborated with a group at Johns Hopkins that worked with the UK Biobank, one of the largest genomic data sets that's out there. We looked at nearly 400,000 patients, and there are two questions that we asked. One, do mitochondrial DNA mutations associate with EGFR, just renal function? There's a spread of renal function in this room. There's a spread in this cohort. Do mutations correlate with EGFR? And then the second question we asked was, do these mutations predict future AKI events? And the answer to both of those questions was strongly positive and independently true. So meaning the more mutations you have, the more changes in mitochondrial DNA, the worse the EGFR was at the time of entry into the trial. And independently, the higher the burden of mutations when you came into the study, the higher the risk of a future AKI event. Okay? And we corrected this second analysis for this first association because we know that CKD is a major risk factor for AKI. And so as we think about this relationship between acute and chronic kidney disease, we think that injury-dependent mutations in mitochondrial DNA may be a new marker of this degradation of information that's happening at the confluence of genetics and environment and aging. And the environment part would be the stressors that our kidneys are encountering all the time. And I've just put up some thoughts here. Could this be a marker of where does the injury accelerate normal aging processes? Kidney cells are long-lived, but they're not the only long-lived cells in the body. And so is something like this possible in other long-lived cells, right? Cardiomyocytes, neurons. How do those cells handle a lifetime of exposures to injury? And so this is the last slide. Hopefully you walk away with acute kidney injury, super common, very morbid, both in the short term as well as the long term, and that we have no treatment in 2025. Second, that these monogenic tubulopathies and other experiments of nature may actually help guide us towards understanding acute kidney injury and its sequelae. NAD augmentation looks like a promising biomarker as well as a potentially promising intervention. And then the stuff we just talked about on mitochondrial DNA. So I will stop here and thank you all once again for the opportunity to be with you today. This is really a deep honor for me and happy to take questions. Thank you.
Well, fantastic talk, Samir. Truly an example of bench to bedside and perfect for medical grand rounds. We have time for a few questions, and I'll just start with one, and that relates to vitamin B3. We have, this concept of renal reserve.
Yep.
And we can even test it in healthy humans or patients. Do you think administration of nicotinamide or vitamin B3 before a renal reserve test would augment that reserve? And if so, in all patients or in a subset, and which subset?
I mean, these are all great questions. And for those who aren't familiar with renal reserve, one of the ways that we test this is by giving a high protein load or a high amino acid load. And what you can see quickly thereafter, like within minutes to a couple of hours, is anywhere from like a 10 to a 40 or even higher % increase in GFR. We have not done that experiment. But what I can tell you is that when we've looked in experimental models, we can see a rapid uptick in GFR. And so I do think that there is something about providing more catalyst to sort of burn more fuel acutely that may change some of the hemodynamics. It's a great question.
Maybe you can collaborate with MediBeacon.
Oh, yeah. That MediBeacon based in St. Louis.
Samir, great talk. That's been fantastic and wonderful work. Related to, I love the data that you showed with aging and these mitochondrial mutations in mice after AKI, and then correlating it with humans as well. I'm wondering, in the mice, if you take it out beyond 1 month, is there any evidence that these mitochondrial DNA mutations go away? And do you have any thoughts about, can we do anything with that?
Yeah, no, absolutely, right. The paper sort of concludes with ways to downstream of these mutations, look at some of the metabolic consequences and intervene on those. But your question is, can we move even more upstream and can we get rid of these mitochondrial DNA mutations? I think it's a very provocative question. It's one that we want to go after. We talked about how the confluence of our inherited genetics and aging and the environment as manifest by aging and injuries may be leading to these somatic mitochondrial DNA mutations. Mitochondria turn over, right? There is this mitophagy phenomenon, and so it does look like if you enhance mitophagy experimentally, you can reduce the rate of mutations. But that's different from targeting the mutations themselves. That's just kind of junking the old batteries and replacing them with new batteries, right? And so there is also an element in here that's sort of like that is very reminiscent of clonal hematopoiesis of indeterminate potential. You wind up with these clones, and what makes those clones persist versus go away? So it's a great question, and trying to work on that right now.
So if I understood correctly, by giving NAD, you can stimulate the mitochondria, eventually doing the ATP on the one hand. On the other hand, there is this concept that if you have more AMP, so less ATP, you may be able to stimulate mitophagy, and that may be also a good thing. So how could we put this together? Maybe it's not much the ATP, but rather stimulating mitochondrial function. What is protective?
Yeah, I think it's a great question, because maybe if I can take it in a slightly different direction. Once you get to NAD as being this thing that you can measure and toggle, right? There are a lot of branch points that come off thereafter. I mean, NAD is involved in probably close to 200 different reactions. We think that the reactions that are, important for oxidative phosphorylation are the ones that are impactful physiologically. But that doesn't rule out the possibility that NAD is also important as a signaling intermediate. Some of the very enzymes are, the AMP kinase, all that stuff you're talking about. Sirtuin enzymes are involved with that. Poly-ADP ribose polymerases use NAD as a substrate. So there's a lot more to unpack there. And that's hopefully why we can keep getting grants to do that. But it does look like different interventions on the mitochondria, at least in experimental systems, lead to benefits physiologically.
Last question, Meg.
This mitochondrial DNA mutation is very fascinating. So mitochondrial DNA is exclusively from the mother side. So these mutations are all induced by the stressor. So some of them are inherited.
Yeah, absolutely right. So we inherit mitochondrial DNA from our moms, right? They come from the oocytes. But then over the course of a lifetime, those mutations are policed by nuclear DNA, like Pol gamma, which is encoded in the nucleus, and other genes that are encoded in the nucleus. And we all share variations there. So there are at least 2 genetic, on-ramps into developing these mutations. And then we have all the environment stuff that we've already talked about. So it's a great point. Thank you.
Samir, thank you so much for.
Thank you. This was great. I appreciate it. Thank you so much.
That was amazing.
I hope it was all right.
That's perfect. I have something.
Oh.